Assalamualaikum to our lecturer, Madam Firuz Maswira. We are going to present our video presentation in the topic High Performance Liquid Chromatography, HPLC. These are the group members, Fatin Dania Aina, Miza Iwani, and Shazwi Shahmi Mairah Najwa. High Performance Liquid Chromatography, or commonly known as HPLC, is an analytical technique used to separate, identify, or quantify each component in a mixture. The mixture is separated using the basic principle of column chromatography and then identified and quantified by spectroscopy. HPLC is thus basically a highly improved form of column liquid chromatography. Instead of a solvent being allowed to drip through a column under gravity, it is forced through under high pressures of up to 400 atmosphere. So these are the example of HPLC block diagram. The purification takes place in a separation column between a stationary and a mobile phase. The stationary phase is a granular material with very small porous particles in a separation column. The mobile phase, on the other hand, is a solvent or solvent mixture which is forced at high pressure through the separation column. Via a valve with a connected sample loop or a small tube of stainless steel, the sample is injected into the mobile phase flow from the pump to the separation column using a syringe. Subsequently, the individual components of the sample migrate through the column at different rates because they are retained to a varying degree by interactions with the stationary phase. After leaving the column, the substances are detected by a suitable detector and passed on as a signal to the HPLC software on the computer.
The main objective of presenting our HPLC experiment is first to determine the rendition time of standard solution, which is the case is caffeine, second to identifying the caffeine peak in a soft drink sample, and finally to calculate the amount of caffeine in that sample using the response factor method.
HPLC is a technique used to separate, identify, and quantify components in a liquid sample. It works by passing the sample through a column packed with stationary phase particles. The components of the sample interact with the stationary and mobile phases differently, causing them to separate as they move through the column. This technique is highly efficient and widely used in fields like drug testing and environmental analysis.
HPLC is widely used in analytical technique for separating, identifying, and quantifying compounds in a mixture, and this is the methodology of HPLC.
Mobile phase reservoir and solvent treatment system. In mobile phase reservoirs, it contains mobile phase solvent with different polarities such as water, methanol, and acetonitrile. The capacity of mobile phase reservoir is from 200 to 1000 ml. It is constructed of glass or stainless steel and contains an exit port open to air. Each reservoir is usually fitted with a gas diffuser, which is helium can be bubble. In solvent treatment system, before reaching the pump, the solvent undergoes the treatment, which includes the filtration to remove particulates using 0.2 or 0.45 nanometer filters, and then degassing to remove dissolved gas like oxygen, which can cause bubble formation and disrupt flow. Degassing can be done by helium sparging, vacuum filtration, or ultrasonication. Proper solvent treatment ensures stable pressure, accurate flow, and consistent detector response. Pumping system. Pumping systems used to force the mobile phase to flow through the packing. Requirement for LC pump is the generation of pressure up to 6000 psi or 414 bar. It must be pulse-free output with flow rates of 0.1 to 10 ml per minute. The flow reproducibility of 0.5% relative and resistance to corrosion by a variety of solvents. There are three types of pumps, which is reciprocating pumps, which is most widely used. displacement pumps, and pneumatic pump.
Next, for the samples injection system, we use two common methods. The first is shrink injection, which is simple and use micro shrink that can handle high pressure up to 1500 psi. The second is the sampling loop method. It is more automated and consistent, typically handling sample volume between 5 to 500 microliters. Next, moving on to the liquid chromatography column. The analytical column is a stainless steel tube, usually 10 to 30 cm in length, and is responsible for separating the compounds. We also use a guard column which protects the analytical column. It helps prevent deactivation by trapping contaminants, and it is also reduce band broadening, extending the column lifespan.
There are two basic types of detector in HPLC. Number one is bulk property detectors that are respond to a mobile phase, and the example of the detectors is effective index detector. And the second one is solid property detectors that is respond to some property of solute such as UV absorbance, fluorescence, or diffusion current that is not possessed by the mobile phase. And the examples of the solid property detectors is fluorescence detectors or UV detector.
Next, I will talk about the result. For the first result for caffeine standard shows the peak of rotation time is 0.849. For soft drink sample run 1 show the peak of rotation time is 0.812. The last one is the soft drink sample run 2 shows the peak of rotation time is 0.825.
Next, we move to data and calculation. At the left side, we have the table of the retention time and peak area of caffeine. So for the calculation, the formula we use for response factor is peak area of compound divided by the concentration. So we got the answer for the response factor is 1131.04. And the concentration of caffeine in soft drink sample in 1:1. The peak area of caffeine divided by the response factor, and we got 4.219 ppm. As for the soft drink sample in 1:2, we divide the peak area of caffeine with the response factor, and we got the answer 12.90 ppm.
This experiment showed us how HPLC systems operate and why each part is critical to getting clear and accurate results. Proper preparation of solvents and careful attention to system pressure and flow are key. We also saw how HPLC is used in real-world applications like pharmaceutical testing and quality control. Through this experiment, we gained practical skills in handling analytical instruments and interpreting chromatographic data.
To conclude, HPLC proved to be an effective technique for separating and identifying compounds. In our experiment, the caffeine peaks helped confirm both the presence and quantity of caffeine in the sample, showing HPLC accuracy and reliability. We also learned how critical factors like mobile phase composition, flow rates, and proper calibration affects the outcome. Even small mistake can lead to big error. Overall, this experiment gave us valuable hands-on experience and show how important HPLC is in field like pharmaceutical, environmental testing, and biochemistry.